Okay, well, thank you everybody for coming back. It's our last meeting. Today is just you. So, and I realize for some of you, perhaps it's a repeat of what you did before. So that's okay. So today, all I have on the agenda, again, we are broadcasting, so I'll let you know that. All I have on the agenda today is to say it's student project presentation. And I'll show our final slide first in case we run out of time, which is I just want to thank you all for being here. I really do appreciate your patience with the snafus of the class and the exploration of the class, and sometimes mistakes in the homework or confusion about things. I appreciate that. NanoHub questions, these kinds of things. I know everybody worked a lot. I know it's a demanding class. I do appreciate the time you put into it. And I appreciate the enthusiasm people put into their projects and ideas. And so I do want to just say to you that I do recognize the effort you put in. And it was a pleasure working with you for the semester. I do want to thank you for the time you put in for that. So I'll do that now before we move on. So who would like to go first for a project presentation? Everybody has to speak, so the order is everyone is I won't say that. Everyone has to speak, so the order is immaterial at some level. Shall I just call on somebody? Would that be better?
I can begin. It's okay. I'll bite the bullet.
Okay. And by the way, people should feel free in this to ask questions. We didn't really do questions and discussion so much last time, but let's treat this a little bit more like it's a little seminar. And so if everyone could try to ask one short question at the end of your classmates' talks, that would probably be appreciated. Doesn't have to be anything very fancy, but that helps you. You can listen to the talk thinking, what question will I ask? Make it the way to focus. Okay, I'll turn it over to you then.
Yeah, I haven't done a lot of changes on the slide per se, so the presentation is not, I was not getting ready for a presentation like if it was a seminar per se, but we made very good progress in combining both of the simulations and making them work. Now the cells respond. I can share my screen here. Just give me a second. Now both of the simulations are running together. This part, it's the same as we saw before. I just organized things a little bit. I think I'll use this as the part of the documentation. Since I can print from a Jupyter notebook to a HTML, I'll use that as the guiding line for the tool when I upload it to NanoHub. That's one of the last things that goes over everything we went on the model. We've seen this before, so I don't want to do it again. But here is a new part that I've been adding, is using now the CC3D representation and telling how, because there is a difference of this computational modeling, which is just representing what is happening, the rates of things to change, and what is happening inside of one cell, and how that replicates to multiple cells on a spatial simulation, right? So here I'm trying to explain a little bit of the challenges of setting up the simulation, which involves thinking about as if it was an experiment that we want to see something. And I explain the reasons for the design of changing strips and how much each strip has in cells, how many cells are in this example. Also letting people know what are the types of cells that we will have in the simulation, or the types of diffusion fields we'll have here, how each of them are generated, how each cell are started. So I say in this Jupyter notebook that every cells are initiated to be healthy cells, and how the cells go from healthy to the other states. And basically creating a guideline or a tutorial for everyone that is playing with this tool. And I also was able to create, it's not here yet, I was still working on it. I was also able to create the steering panels where the user can change things. And basically what the user will control is how much the cells generate virus. On the first state, there's two stages that the virus can be created. One is when the cell bursts and dies. So there is a release of virus that was infected by viral load first, because this model actually is trying to replicate something we see, which is some cells gets infected by virus, they undergo paraptosis, they let their neighbors know that they're going under paraptosis because they're infected, and these neighbors on its turn initiate paraptosis. Paraptosis has a benefit of letting the immune system know that something is wrong in this area and requiring more immune cells. And in a primitive way, acts almost like a control burn or something that we see in some spread wildfires, which we're trying to contain fire with fire, basically, right? We burn an area. So when the fire gets there, there's nothing to burn and the fire goes away. So the same thing, similar thing happens here, where if the cells around start paraptosis, there is nothing for the virus to replicate because all the cells are already initiated this stage of dying. So there is benefits on this in the case of like avoiding widespread to the virus, but it also causes a lot of damage in the process if it's not controlled. This model doesn't have anything that controls the paraptosis, like turning it off, but it's something that we intended to put in the simulation to replicate what we see and see how much of the immune response is necessary to control it down, because we know from some literature that that's one of the main ways of cells to stop going paraptosis is immune cell coming in and basically taking away all the IL-1 beta that's being created and stop the paraptosis to overtake the whole body. But yeah, that's the how much I could get. I'm very happy. I can show the simulation again because now it's running with the correct numbers. I know oh, sorry about that. I know that the last time we talked and do the presentations, we were not saying the same things we said before. So I'm trying to just update everyone on where the simulation is. So these are default values. It's all arbitrary, but we start to see the number of cells dying by paraptosis instead of the viral load, and how much the viral load actually spread by looking at these two fields. And here we can initiate with different values. One is viral production when the cell is infected. So the cell will release a number of virus at every Monte Carlo steps. And the other one is just a burst when the cell dies. One of the things that I noticed just playing with this is that the burst has less significance in this case with paraptosis than the actual production of virus at every Monte Carlo step. I pressed pause and now my thing crashed. But yeah, this is something that the user will see it can play around with these values and see different results. Some of them, for example, if the viral production is higher, we'll see that a lot of cells die super fast and start replicating more virus, and the pyroptosis is basically negligible, the effect that it can have on the whole system. But yeah.
So I have a question about that. So if you make the levels of viral production low enough, did you ever have a point where the virus doesn't spread, where this tissue
Yeah.
doesn't completely die?
Yes.
Could you
Right
show,
now,
could you show
yeah, I
that?
can show. Just it just crashed. I'm reinitiating it.
So you have three regimes. One where the virus, where the interferon shuts down the viral production, and there's some uninfected cells. One where the virus spreads, but the infection, but the pyroptosis gets ahead of it, and a third one where the virus spreads so fast the pyroptosis does nothing.
Yes.
Distinct regimes.
Yeah. So here we'll put the viral production even lower than this. This is the minimal that I had set, because it's when we lower than this, we start to see the same kind of behavior. So it doesn't change a lot of things. So here we're just accounting for the burst, and we start noticing, and here on the graph, we can see too that like now no cell will die again because of viral production or reproduction of virus, but all of them will eventually die because of pyroptosis is uncontrollable here. If I just set for let me do a one step here. If I set this low and set the burst very high, we can see some viral reproduction happening. You see it's fast, but it's less effective than just having virus being produced constantly by the cell. But as you could see, there was one cell here that turned green, but now basically all of them will just die by pyroptosis. And the value here is very large if we compare, but since it just happened when the cell dies, it's also understandable. Here with different values, we can have like an equilibrium when cells die at the same rate. If I'm not wrong, was 60. They die at the same rate for virus and for pyroptosis, or I don't think it's this one. Yeah, 60 is too much. You see, the virus is overtaking, and basically no cell die by IL-1 beta or pyroptosis induced. It's very low, the number. But there is a correct number here where it's it reaches an equilibrium. I just don't remember it from the top of my head.
So if you controlled the diffusion length of the IL-1 beta by changing the decay rate, so you made the IL-1 beta not diffuse, you could cause it to diffuse less. Presumably then it'll be a less effective signal. I wonder if that would be a nice thing to include in your eventual.
Yes, I was thinking about that with that conversation we have about the Einstein calculations for diffusion. Since the molecule is way smaller than a virus, the virus is small, but the molecule is way smaller. I would assume that the spread of the molecule is faster than the spread of the virus. That's something that I want to put in the model too, but it's something that I haven't done yet.
But the diffusion length depends on the diffusion constant and the decay rate. So if the virus lasts longer than the molecule, the virus can still get further, even if it goes more slowly.
Oh yeah, it's true, right? If the decay happens.
So it might be interesting to try tuning the decay as a user controllable parameter as well. But
Okay.
already that you see these different regimes is interesting. I think it's quite impressive.
Yeah, here we start to see a little bit of a equilibrium between how many cells die by virus and with the production a little bit bigger, right? I don't have the biology of most of you guys, but I noticed that there are virus that they reproduce differently in the sense of some cells just release when they burst, some cells just release them constantly. So the general idea here was to create a way of like mimic both worlds with one simulation.
Yeah, I mean, it varies a lot. In general, I mean, flu and COVID both, the belief is that usually when the cell dies, it doesn't release much viable virus, because the virus has to be assembled.
They need the membrane, part of the membrane, right?
They need the membrane, and so the cell has to be functional to be able to assemble the virus. Now, it's certainly true that there are definitely, especially, for example, for phage, for viruses that infect bacteria, they typically are lytic viruses. They build up inside the bacteria until the bacteria bursts, and then they're released all at once. And so it's interesting to see the difference in effect that pulses versus the continuous.
Yeah.
Well, Jackson, I'm sorry you're not not able to use your voice today, but get better for it. Any other questions from the audience about this? Suggestions? This is promising, but what could Joel and Jackson do to improve their demonstration, do you think?
I don't have a question on that, just on like, because I know you're trying to implement the original paper. What would how much more work and how much would it change your simulation to implement all those different depth variations?
That's a good question. The problem is a lot of the same molecules that are present on their paper is present in different types of cell death. And the decision making is the most challenging thing, and that's one of the things that I think I will, dedicate my time on my PhD is like, what makes the cell decide which type of death they undergo? Is there one factor, multiple factors, a combination of factors? So it's a open-ended, question. But yes, for example, we try to do the necroptosis model too, and the idea was to add into a single module of cell death. But we see some molecules that are here in this proptosis model. It's also in the other one. And it would be interesting to see how they would react if we load both of them in the cell at the same time. Because right now, for example, right now, the cell here decides on quotes which type they will become, if they become pyro IL-1 beta or DAMPs, just by the amount of the diffusion field is seen by the cell. So it's imitating a toll-like receptor. Since both, I have to read this, I don't know if this is correct, but since I'm assuming that both molecules bind to the same receptor, the one that binds first a specific amount, then the cell undergo a type of death. But it's hard to define where the decision happens in the biological.
Best model
It's a great question.
saw just randomly decided. I just did like a random number generator basically to decide what cell death pathway it went down.
I think Hoffman's paper tries to dig into that in more detail, but it's pretty messy.
Yes.
I mean, this is the problem you face with all these biological control phenomena. which is, as Joel said, and Jackson would be an expert, you have a lot of different outcomes. You could have cell death through these different pathways. Upstream, you have a series of receptors that detect certain events or situations. And then you have a sort of a hairball of connections, which are both activating and inhibitory, which pass to the different possible death pathways. So cells are always making a decision about whether to grow, to survive, or to die. Within those, there are different ways they can grow, survive, and die. So it's not just a single set of decisions. But cells are always integrating a series of signals, some of which are promoting cell growth, some of which are promoting stasis, some of which are promoting death. And the way those signals are integrated is complicated and still imperfectly understood. But one of the differences between humans, chickens, and bats is that some of the cell death pathway readouts are different in the different species. So that the same signal through this toll-like receptor leads to a different kind of cell death or no cell death in one species versus another. And so you can imagine, from a sort of a computational point of view, you have some sort of API, which is your toll-like receptors. They have inputs on one side, they send outputs on the other side. You have some set of downstream effectors, which have their own API. And then you have this complex wiring diagram connecting the outputs here to the inputs here. And it's different in different species, which seems strange. The reality, of course, is that our abstraction of molecular circuits in cells is just that. It's an abstraction. In reality, every molecule in a cell reacts at some level with every other molecule. The ones that we single out are the ones that react most strongly, or the ones that are identified as being important. But that doesn't mean that those other reactions don't exist. And so that means evolution can work on that. You get a little bit of the same kind of thing in vision, which is if our photoreceptors were absolutely fine-tuned to a single frequency, then we wouldn't be able to do the kind of color perception that we do. The fact that they're tuned over a broad range and that the sensitivities overlap allows you to do interpolation, and it gives you plasticity, something for evolution to work with. And so the fact that the signals aren't perfect actually is something that allows the system to be more adaptive than a very finely tuned system might be. It's a sort of antithetical to computing. In computing, we have to have an API that's very well defined. You want this to connect to this. You don't have this connect fuzzily to a bunch of things. Of course, these days with neural networks and artificial intelligence, we are a little bit more familiar with that kind of fuzzy interconnection. But biology takes those things to an extreme in many systems. Sometimes you get what's called catalyzation, where you in fact get something that looks like a real distinct separate outcomes. But most of these biological decisions are pretty poorly understood. A classic example of this, which would be simpler in some ways, would be, say, a bacterium, which will swim towards something it likes and away from something that it doesn't like. And so it'll have chemical sensors on its surface detecting the concentration of glucose, the concentration of ammonia, which is a toxin, the concentration of other molecules, some of which are attractive and some of which are repulsive. Ultimately, the bacterium only has one output, which is, do I swim towards it or away from it? And so what happens, what does the bacterium do if it faces, gee, there's a lot of sugar here, which I want, but also a lot of ammonia, which I don't want? how is the decision made? And there's been quite a bit of work done on that, but I think there's still a lot that's not understood at the molecular level about how single integration of decisions are made. So that's a sort of a long-winded comment about that. But I think what you asked is a very profound question. And it does get at some of the difficulty of unraveling biological systems and the lack of clarity because they're not designed. They're not designed to be intelligible. They're not designed at all. And so again, as I've said from the beginning of this class, things like orthogonality, which are such basic design principles in engineering and computing, are not present in biology. You have, in particular, molecules that have structural function and signaling function. Now, in a classic engineering system, you'd separate those two functions. Now, these days, with embedded systems, you're moving back towards something more biological, where we had somebody who gave a seminar here in the Intelligent Systems Engineering a few weeks ago, who was putting sensors into bricks, sensors into wood, and actually trying to make the sensors not just out of, you take a small microprocessor or cell phone and you embed it in the wood, but actually trying to make the sensors out of the material itself, so that the distinction between the sensor and the object was blurred. That's a very bio-inspired strategy. And it can be very powerful, but it's hard to design because we're not used to thinking about problems that way. It makes it hard to understand how the system will behave. So I think that was long-winded, but I think it's a question that's important. And, that's one reason this kind of agent-based modeling can be so powerful, because until you see the system in action, you don't really get a sense of how it's going to behave. Just watching the spread of the chemicals here is fascinating. The patterns of chemical spread. As you change those parameters, the spread is very different. In the end, the outcome may always be that all the cells die, but the order in which it went, the relative significance of the cell death pathways, the temporal sequences, these are very important things to understand. Some years I give talks on what I call qualitative, semi-quantitative, and quantitative analysis, which again seems very abstract. But the order in which things happen can be very important. Whether something is bigger than something else may be important, even if the exact values aren't important. And you saw that here. In some situations, death through IL-1 beta was very strong. In others, DAMP was strong. In others, both were strong. In others, neither was strong. And so those different outcomes, what I call semi-quantitative, because it's not the exact value, it's the relative values that matter, can be quite significant. Great. Any other questions or comments?
So really quickly, you mentioned very briefly that typically there would be a feedback mechanism to prevent the case of it just being complete cell death once the virus is contained. I was just curious what you thought it would take, both in terms of like effort and added parts, to implement that from what you have right now.
Yeah, we would have to have some other type of cell showing up. In this case, would be an immune cell. I don't know which type. I would have to do a little bit more research to see which one responded to a pyroptotic event and see how they, my knowledge in biology is a little bit limited, but we know that something controls it, because otherwise every time a cell would go under pyroptosis, we would all die. And since it also works, the main focus of a pyroptosis is to activate the immune system or request help from the immune system. what tells me that something is connected with that. You know, the immune system will do the cleaning, or will tell the cells to like don't, or it cleans the amount of IL-1 beta in the environment, or it will just prevent the cells to have the signal to enter in them, right? And they start the pyroptosis by themselves. So it's one of the two. At least that's what I have in my head right now without reading anything in particular. But that's a good question. It seems simple to add just another cell here, but the mechanism behind it, like how much it takes in from the IL-1 beta and stuff like that could be tested. Actually, it would be a good test just to set up those parameters if we cannot find it in any literature. But yeah, basically that. Just add another cell. If you remember, there is this model from Giuliano that and was also Josh, right, Glazier, that they did that one that cells actually shows up out of nowhere in the simulation, and those cells are representing immune cells, and they're acting upon request, basically. So the same kind of simulation would have to happen here. If I'm not wrong, if I'm wrong, please correct me, Dr. Glazier.
Well, I mean, there are a lot of different kinds of shutdown mechanisms you can have. There is a pattern which you see in biology a lot, not just with respect to the immune system, which can seem sort of confusing the first time you see it, which is that when you have a signal, that signal will turn on two signals downstream, one of which suppose you go from initial signal to an intermediate state to a final output. This first signal will turn on two things. Let's call it A turns on 1 and 2. 1 turns on B and 2 turns off B. What's called an incoherent feed-forward now. And the question is, why would you have the first signal produce something that both promotes and turns off the target? And the answer is, if you're only thinking about steady states, that's not an interesting thing to do. But if you want to create an interesting temporal response, it is an interesting thing to do. Because what you do is you turn on the activator quickly, and you turn on the inhibitor slowly. And then you have a pulse generator. So you turn on IL-2, that's pro-inflammatory, fast. And then you turn on IL-10, that's anti-inflammatory, but you turn it on more slowly. And so then you get fast buildup of your activated signal. The same thing turns on. That activates slowly the second thing, but then it turns off the signal that it started. And so the immune system is full of feedback loops like that, where the same signal turns on something that will turn on the immune response and turn it off. And typically the turn off is done slower than the turn on. So the turn on happens quickly, it has its time, and then it's turned off. The problem then is if you have additional damage, you're always in that turn on phase, and then you can get runaway. And so I think it's worth taking a look, Joel, at certainly there are cellular turn off responses too, but there's certainly molecular ones. So you might look at a basic, again, Wikipedia will do this. You look at the basic feedback loops for interleukins. You'll find that there are fewer anti-inflammatory ones than pro-inflammatory ones, but there are a few that are anti-inflammatory. And they tend to be turned on slower and kept on longer than the pro-inflammatory ones. And so that pattern happens quite a bit. Pulse generation is something that biology is good at. You create an initial stimulus, it generates a pulse and then turns itself off. And that's a sort of a fundamental organizing principle. We talked very little about motifs. That's a classic motif. And it requires what's called an incoherent system, where you have both, you turn on signals that fight each other, which again is something that typically we don't do very much in engineering, although systems with feedback and control sometimes will do that. That's a great question. Okay, any other final questions before we move on to our next presentation? Well, that's great. I appreciate the people asking the question. And I appreciate seeing the demonstration. Okay, who would like to go next? How about Josh? You there?
Yeah, just give me 1 second.
Okay. Some years I do a formal sign-up list, and then 4 groups. It should be on it.
All right, can everyone see that?
Before I start, is your name Lithuania?
It is, yeah.
Okay. I've been looking at it for the whole semester and wanted to ask, and I thought it maybe be rude to ask. But I couldn't resist the last class to ask that question.
Oh no, yeah, you're fine. No, they immigrated at the start of World War II when Hitler invaded Poland and came to Ellis Island. So, but yeah.
My father was a migration researcher.
Oh, cool. Oh, okay. We maybe talk about that later, but I'll go ahead and get started. So my project was on recreating / updating a previous work called Emergent Networks, a slime mold simulation. I have the link to the paper here. It was a bachelor's thesis completed in 2011. So we'll briefly go over an outline. So I'll start by just defining the goals of this project, the underlying biology, kind of the high level mathematical model that I will use when I go to implement it using CompuCell 3D. And then we will go through replicating the results of the original paper. I also replicated some other results from other things I found, other sources and experiments I found online. Then we'll just go through some main takeaways in a brief demo. So the main goal was to replicate the results of the previous paper. Like I said, it was called Emergent Networks by Nicholas Simler. The main challenge with this aspect was that since it was done in 2011, at the time, the latest version release of CompuCell was 3.x or 3.3.x. It is now 3.4.x, and apparently a lot of change happened with that update. So originally the model wouldn't run at all, so I had to do some code wrangling. But in addition to updating the model and getting it running again on the newer version of CompuCell, I wanted to turn it into a publicly available education tool to upload to NanoHub. And a part of this was adding the steering panel, which is essentially a graphical user interface. And this would allow programmers and non-programmers alike to kind of fiddle with the model parameters and see how it affects the output of the simulation. So now going into the underlying biology of slime molds, first, there's over 700 unique species of known slime molds right now. They're heterotrophic organisms, and that essentially means that they can't make their own food. So the way that they consume nutrients is this process called phagocytosis. And essentially they just consume the food source and then kind of just go through their own process of digestion. It's its own biological process that's not necessarily relevant to this actual simulation, but I thought I'd mention it. And so, like I said, there's over 700 unique species, but there's two main types of slime molds. There's the plasmodial and the cellular. The main difference is the plasmodial is an enormous single cell with thousands of nuclei, and the cellular are kind of individual cells aggregated into a great swarm. And there's two big groups of cellular slime molds, the dictyostelids and the acrosadia. I'm sorry, I can't pronounce any of these names, but we're primarily going to be talking about cellular slime molds. So here's a brief figure of kind of the life cycle of the cellular slime mold. So it starts off with this meiosis, and then there's this germination kind of period, and then it kind of goes through this, goes into a zygotic state, and then undergoes mitosis. And then this is the main part of this life cycle that we're going to be talking about, or simulating rather, is this plasmodium stage. And it goes through the haploid and diploid cycles. So this plasmodium stage is in the diploid part of this life cycle. So we'll just continue on with the biology. So like I was saying, the main thing that we're focusing on is the plasmodium stage of the cellular slime mold. So during this stage, the slime mold is an amoeba-like organism with a broad tubular network. The main computation of this biological system is to create networks for communication and transportation, namely the transportation of nutrients and the communication of kind of where food sources are, as well as where toxins or other obstacles to be afforded. And so this is kind of a two-part system. When it creates this network, it creates these actin-myosin fibers, and then it passively transports the chemicals in food in these kind of sinusoidal waves through the fibers, and the direction of the fibers limit the flow of this plasmodium. And to kind of transport these, they develop long tubes, these long, thin, open tubes. However, they are bounded by an upper bound, and kind of a general rule is that the more plasmodium that flows through the tube, the larger the tube is. So that's the underlying biology. So now we'll go into how we actually model this into a computational simulation. So there's three main parts: direction, structure, and optimization. So direction, where will the slime mold go? The main kind of factor in this is local inhibition and long-term activation. And so the way we model this is with attraction and repulsion via chemical fields. So the slime mold will be intuitively attracted to food sources and repulsed by obstacles or unfavorable conditions. Now, specifically, I didn't model unfavorable conditions, but in experiment, they've been known to like steer away from, cold temperatures or too hot temperatures. I just modeled just a simple obstacle, like a barrier, a wall, something of that nature. But an extension to this could be, maybe a temperature parameter or anything like that. But kind of the idea of this local inhibition, long-term activation, is that the attractants will have high diffusion rates and low decay rates, and the repulsive ones will have low diffusion rates and high decay rates. And so kind of once the slime mold is attracted to something, how does it move? So the slime mold moves with an autocatalytic loop. The main thing that drives this is the connectivity plugin. And essentially, this just penalizes blob-like structures. So like in the previous slide, the goal is to create this tubular structure that optimizes this flow of nutrients and other transmitters that allow it to create these networks. And so we implement that with a high medium adhesion and low cell cohesion, and this incentivizes the slime mold to explore. And also the cells release additional attractants to kind of keep this going. And as far as and then after it is moving in the correct direction, it has to optimize this tubular network so that it doesn't, waste resources. And so how does the slime mold construct the optimal pathways? And that's through growth and oscillation. And this is modeled through an increase and decrease in the volume of the slime mold. Next, kind of the assumptions and things that we had to make is that all chemicals saturate at a specified level. And this is to mirror the finite molecular size. And food sources increase attractor secretions at fixed intervals. And this is to appear less homogeneous. And kind of going into the details, the main kind of driving force is the chemical fields. And so this delta E sub chem is the change in energy for the chemotaxis plugin in Computational 3D. And this essentially tells us how much the energy will change based on three things mainly: the lambda, which is mainly the overall strength of the chemotaxis. And then that's dependent on this function C, which is the concentration at a point X. And S is this saturation linear coefficient. And then kind of going into how the concentration is distributed throughout the field, it's modeled in this partial differential equation, partial C, partial T. And so it's dependent on alpha. These two parameters, alpha and epsilon, are two constant values, and they're multiplied by this Kronecker. It's the Kronecker delta, and that kind of limits the amount that is secreted or decayed in the chemical field. It is also dependent on the D, which is the diffusion matrix, as well as the concentration of the field. Now going into implementation, how I actually coded this up or how Nicholas and then me porting it over to CompuCell actually implemented this. Briefly going through the plugins and steppables, the usual ones, volume, local flex, center of mass, and neighbor tracker are all pretty, and contact are all pretty standard. Connectivity this ensures that the tubes that form stay connected without a high. This essentially creates a large penalty if the slime mold kind of breaks up into sparse, unconnected networks. And then chemotaxis and secretion are the two main plugins because, like I said, the main thing is the chemical fields in this simulation. And then as far as steppables, there's diffusion, decay, and secretion, and those go into the chemical field as well. So one of the main things that were implemented was this growth, and that was for the optimization. That's how the growth or the optimization was modeled. And so there are these two constants. There's the base volume of the slime mold. This is just an arbitrary constant. The change in growth per unit time, that's dependent on the base volume, but I just gave an example value for this slide. And so there's two kind of helper functions. There's this volume dev, which calculates the amount of volume that the slime mold will either grow or shrink. Obviously, a positive is a growth. A positive value is a growth, and a negative would be it shrinking. And then also you have to find the number of connected food sources. That also determines how much volume that you need to add to the slime mold. And so this is just a code snippet from the actual steppables.py file from the simulation that I will show running in just a second. And then this is the oscillation of the slime mold. So there are these the percentage of volume that is oscillating and this is a sinusoidal function that is calculated every or every Monte Carlo step. And then this is kind of everything all together. This is the step function from the main kind of volume steppable. That was a custom steppable. And so it essentially checks if the growth period is over, because it goes over these kind of periods of growth and exploration. And then we're just implementing all of the two methods that I just shown. And then finally, you just update the self.slimy is the slime mold cell type. So now we'll go into the replicated results. So first is the similar paper, the original kind of work. So one of them was the 4 food sources with no barriers. So I think it was kind of hard to replicate because a lot of these, he didn't give any parameters for like the lambdas or anything. All I went off of was what he used in the code last that I was able to get a hold of. So a lot of these I don't look a lot alike, but I think this, like for instance, I think the main factor was just how many Monte Carlo steps he let it run, because I think some of them were only 10,000, whereas some of them were upwards of 30,000. But I think this one, he did actually, I think this was around 10,000 Monte Carlo steps. And so I was able to do it, but I think the parameters were off because it's around the same structure, but there's a lot of these kind of little extra kind of networks. It needs to be a lot more kind of less or more sparse. But I think that was just, I couldn't find the correct parameters that I used. But you'll see in some of the other ones that I was able to find some pretty good ones. So this is 4 food sources with barriers. So very similar to this one, but you can see it's a lot more centralized because it wants to get away from these blue barriers right here. Sorry, it's my presentation is being very, okay. Sorry, it just wasn't updating for some reason. And then another one was 3 food sources with different initial conditions. And really the initial condition is the starting place, the X and Y coordinates of the slime mold. And so the first one was it starting off in the center, and the other one was starting at a food source. These look very similar. So I think the parameter values were fairly decent. But I think these big loops could be, kind of pruned if I let the simulation run longer. But these were the instances where he said how many Monte Carlo steps he ran them for. But again, I'm not sure if the other parameters like the lambda values and everything was correct. All right, and then this is the large network. Again, he cited some of the papers that I was interested in, like the Tokyo Railroad Network, but he never really specified which one was which. So I just titled this large network. It looks very, very similar. I think maybe the parameters that he was using and the code that he sent me were from this because it looks very similar. He also, you'll notice that this red line, he actually drew another kind of just a very, very thin food source to connecting this other one to get that. But I didn't do that, but it looks very similar. I was happy with this result. And then kind of to extend it, I found a random GIF online kind of showing these 2 food sources in this maze. And you'll see it traces out this path, and it's pretty decent. I just couldn't, I think I just need to let it run longer so it can kind of prune this big path out there. But I was kind of ran out of time, but I was pretty, it found one of the shortest paths, and it was starting, this was for example, this was all the way down here and it started shrinking at some point. So now I'll just go into the quick demo. So I'll very quickly switch over to so unfortunately, it still won't let me just share my desktop. So I'll just have to share the simulation volume, which is unfortunate because I got my steering panel to work fairly well, including the connectivity. But I guess I'll just, oh. Wait.
There you go.
Okay, there it goes. So this is just the 3 food source one. So I can go ahead and let it run. Like I said, the connectivity is essentially just, it'll penalize if there's kind of breaking up. So if we set that to 0, it'll go ahead and start doing that, and then set it back up to there. It'll start connecting again. Can mess with the lambda values. So like, for instance, if we, it's all these blue it's repulsed to the blue. So if we create that, it'll start being attracted to the walls, everything, and then can change all the saturation linear coefficients and stuff like that. So then you'll see as it goes, it kind of creates these extra kind of circles, and then as it'll go, it will slowly prune them down to these. So
I have a question. Can you show us the diffusion fields actually happening? And just to see how it looks.
yeah, so they're it's a very, very slow, but it is continuous. And then you can see this is the, so this doesn't really diffuse at all. But so.
So does what you just showed, was that a chemical field belonging to the wall?
Yeah, this is just the repulsive ones. And so the only cell type with this is the wall, but I could very briefly just to show one with more barriers. So this is the one with the extra barriers. So it has the wall as well as the other cell types with the repulsive diffusive field. All right, so that's pretty much it for the demo, unless you want to see anything else, or are there any other general questions?
Yeah, I have a question on the demo.
Yeah.
Kind of thing. So all the green bits, is that just one giant cell that's
Yes.
kind of stretching itself out?
Yeah. There's only one slime mold type defined in the PIF that this is initialized with.
Is that how mold works in the real world? I'm not really. Is it just one giant cell that connects to everything? Or is there some kind of delineation at some point?
Well, it depends. We had to make a lot of kind of assumptions, but there's two types of slime molds. And like plasmodial ones, it is just one giant single cell with thousands of nuclei. But the cellular one, they are individual cells that aggregate, but I'm not sure. I think it might have been some computational things why they didn't, use mitosis to, model that. I think it'd be easier just to, because essentially what's happening is it's just as it's going, it's modifying the target volume of the slime mold cell type. And then it has a really low, like if I go to the contact, this is the one, or food source and medium, or medium and slime mold. So it has this really low or somewhat high adhesion to it. That's kind of the main function of that.
Other questions?
I just have one small follow-up one. So since the obstacles, their chemical doesn't diffuse, is there a reason why the original model said that they used that instead of just changing the contact energy, or would those not do the same thing, just like in terms of making sure it sticks or doesn't stick to the obstacles?
I think that it has to kind of have this diffusion so that it won't be attracted to it. Like not necessarily so that like I think the contact is so that if it does touch it, which sometimes it does touch it while it's kind of permeating the space. It's just that once it does touch it, it doesn't, have any reason to get stuck to it. So I think the diffusive field is so that it won't be attracted to it. And then the contact energy is so that once it touches it, it doesn't stick to it.
Okay.
That's my understanding, at least.
The diffusion then you set up is just diffusion inside the cell for the blue ones? How did you define?
I believe so. So yeah, it's just there's only one secretor, and that's the attractor.
Yeah, because I was also thinking the same way as Connor about, I thought you were playing with the contact energy when you were repulsing it from the walls or the blue squares.
No, I don't. Nothing happens to the contact energy throughout the simulation, unless you physically go into the model editor and.
Cool.
I guess I misunderstood too. I thought the repulsive field diffused.
I mean, it does, but it has a very, very low.
Okay, so it diffuses, but with a low diffusion.
Yeah, so like a pretty
okay,
low diffusion constant and a really high decay constant. That was kind of the modeling of the local local inhibition.
Right, got it. So Connor, so it is diffusing, it's just not diffusing very far.
Yeah, that's where I got confused.
Okay. Because that also, and then the contact energy with medium causes it to want to minimize its total surface area, right? Because the more surface you have, the more costly it is. If you make that number too big, then it would break up into blobs. And so that would be something you could actually steer. If you make that contact energy with medium too low, then it'll spread out everywhere because there's no cost to spreading. If you make that energy too high, you'll break up into blobs. So that would be something that potentially could be added to it.
Okay, yeah, because there was so many parameters, I wasn't sure which ones to steer, so I just kind of picked
Right.
one. But they're really easy to add, so yeah, I could definitely add that one.
And then, in principle, you could have something that's sort of a feudal loop at the beginning where it loads the initial pattern, which pattern you want to use, pull down menu for that.
Mhm.
Not that you haven't done enough already. I mean, it's very nice, but I'm always thinking about ways of improving it. I think the one thing I noticed, which was just a small thing, you said in a couple of slides it was the cellular slime mold,
Mhm.
but the plasmodium is for the acellular slime molds. So
Oh,
the slide where you said cellular, you meant acellular.
okay. Yeah,
It's,
sorry.
it's a bit confusing. It's the terminology is a bit confusing. In fact, people, if you look at Wikipedia, you'll find that I put the links in the chat. That there was a lot of confusion historically about slime molds because they're unrelated species that were grouped together. The difference between the cellular and acellular slime mold was confused. But if it's a plasmodium, it's the acellular slime mold.
Okay. Yeah, I misunderstood, and I'm not a biology person, so
That's fine. But just so and again I put the reference the Wikipedia link in the chat there too. But that was the one thing that the description of what they did was right, but just the heading, the title was backwards.
Okay.
It's easy to.
Right. Thank you.
No, that's fine. But that's very nice. The optimization is very interesting. The fact that it does this finding is quite nice. And I think if you turn off the oscillations, it doesn't work. If you make the oscillation, I think that's another thing. If you make the oscillation amplitude too big, again, it breaks up. If you make the oscillation amplitude too small, it finds a pattern and then it never optimizes. So that's another one that's quite interesting. There's a lot you could do. This is a semester project. It's not a lifetime project. So you've done a lot, but there's a lot one could do with this to take this and go further with it. I think it's quite intriguing. It was a great choice of a source paper, even if there were some inadequacies on the part. That must have been, I mean, that the person who did the original work as an undergraduate project, it's pretty impressive.
Yeah, definitely.
So I think this is great. Other questions before we move on? I think it's a great demo. It'd be good to have that. Have you tried putting it up on NanoHub yet?
No, not yet. I was waiting. I still wanted to. I wanted to add the PIF initializer to the steering panel, but I was running into some issues and needed to get my presentation done, so I kind of put that off. But I'll work on that now. And then once I get that figured out, because I don't want anyone to have to edit any of the code at all, unless they want and know how to. So I just want to. I don't think I'll be able to get like one that will generate like a random one or anything like that, but I think I would definitely, in the documentation layout, here are the ones that exist. You can choose them and then you can create your own. But I think the ones that I have are pretty sufficient, I think, just for like a general learning tool.
I mean, the control panel, I mean, the steering panel has the option of a pull down menu.
Yeah,
So
that's what I was going to do.
You could list the files you have and ask which one you want to read in at the beginning.
I think the thing I was confused on was I was going to try that, but I don't, because I process all the steering panel data in the step function. And so if you started the simulation and then wanted to change it, you'd have to like restart the simulation. But then, if I initialize it, I was kind of having trouble with that. But
okay, because you're using you're reading the PIF file in through the in the XML.
Yeah, so kind of the way I do it is that everything is defined in the XML, and then I have IDs for everything that I want to steer that I defined in the XML, and then I just grab it in Python and update it.
Yeah, so for that one, you'd have to actually move the initial, the drawing, the configuration into not even the start function, it would have to be the step function.
Okay.
And then I think Connor, we did that. We did that looping. We showed how to do that. We worked on that, right? We looked at the loop to do a loop for it start to set up the simulation.
Yeah, we did. We ended up doing something a little bit different in our code, but we talked a little bit about that.
Right. So if you want, I mean, again, this is plenty, but if you want to try that, Josh, we could just send me an email. We can sit down and I'll show you how to do it. It's not hard, but it does take a few extra minutes. We have to ask Maciek probably. I don't know whether you have a Python way of reading the PIF file in and turning it into an initial condition. There must be a method to do that, but I don't know what the command is. So that would be a good question.
Okay.
About how to do it. I mean, I know how to do it by hand, but but there should be the same thing that the code uses when it reads the XML file should be available in the Python to do it.
Mm-hmm.
And so let me send an email. Okay, great. Sorry, I just wanted to send that query out immediately so that we didn't. Something that I really should have done early in the class, but I don't tend to use it. So my mistake is there is a Reddit forum for questions like that for Computational. And I guess I'm spoiled because I have the people who develop it next door, so I don't have to post to the Reddit forum. I just ask TJ. But in fact, I should have sent you all to that too, because there is some hive mind. In other words, sometimes we'll post a question there and somebody we don't know will answer it. So that's something for next year. I'll try to make that a little bit clearer. Thank you. Great. Okay, who's next? Connor, I guess.
Yeah, we can go.
Okay, I think Alex was the first last time, so I guess we'll reverse the order.
Only fitting. So going to start off with a little bit of a disclaimer. We had some technical difficulties while trying to get to where we wanted to be for today. So at the end of our last presentation, we finished the statement that the simulation that we had programmed would take just over 6 years to finish running. So Andrew and I very quickly pivoted and started working on a different simulation that we could finish before the end of class, and then talk to Professor Glazier about things that we could do to get the larger simulation working after the deadlines. So we literally managed to get it close to where we wanted with the technical difficulties getting our way about 45 minutes before class today. So there's not much to be shown in terms of new presentation slides, but I can walk you through a little bit of what happened. So basically, earlier in the semester, Can everyone see my, okay. So this is a slide from our older presentation. And we had made a single cell chemotaxing up a noisy gradient as kind of like our intro into chemotaxis and punching holes in the gradient and all of that fun stuff. It's not a great simulation. There's a bunch of things off about it because we were still learning chemotaxis, and also, so this was the field from the paper we were trying to replicate. And as you can see, it doesn't actually ever dip down fully to 0 except for where there are holes punched into it. And the way that we did this simulation was that it had holes punched into it at increasing frequency as you moved away. So there were some things to change. And another thing we wanted to change, since we were no longer trying to replicate an exact paper, is that Joel made the comment last week, which is it really that realistic that you would have all these random holes of 0 chemical gradient throughout the field? And the answer to that is no. So what we did instead was we made it so that the dips were evenly distributed, but how much the field dipped in terms of noise was divided by a factor of 1.2. So it was slightly more realistic in that sense. So what I can do now is that I can show you, can everyone see player? No, okay, let me stop sharing and reshare then. How about now? Okay, so what we basically did is that we have here a smoother gradient like the one in the paper with the dips that I just described, and here is the cell on the right. And then we have a plot for the cell's trajectory over time. And we also calculated the speed along the x-axis, the y-axis, and then the total speed. And the way that the simulation works is that you select what you want your initial conditions to be in terms of the volume and the noise, and then you pick which one of those is your variable. And what it does is that it holds the one constant and then it scans along the other one. So the lowest we have is 25, and I will set the noise for 1% and say that I want a very long volume because that's faster in this case. And every 2,000 steps, it ends up increasing the size. And also, this is actually a very rare circumstance right now. There's a bunch of clustered noise right here that's gotten anchored on, which doesn't happen very often when you're simulating it randomly. So we can always poke it again. I'm actually going to go ahead and restart it. That's very unfortunate that that's how it initialized that time.
True random.
Yeah, no, it really is. Okay. Yeah, there we go. So as you can see, it's kind of moving all around up and down. And what's going to happen is that when it gets within a certain distance of the focal point, it changes the season. Now, what's interesting is that we were talking earlier in the semester that when the cell is larger, it should do better. But it's actually the case, at least for the parameters that we have, is that if you're really tiny and there's large enough gaps between the holes in the gradient, you just kind of move around them and ignore them. But as you grow larger, you end up having to confront more noise in your environment, which can be confusing. So there's actually a sense in which, assuming that the noise is small enough, your speed is actually higher because you can just kind of ignore all the patches. But once you're large, it gets to the point where you can encounter walls of them. And depending on your size, it could end up being such that the walls spread along your surface and none of the gaps are enough for you to fit through.
That's interesting because it really doesn't want to go over a gap. It doesn't want to go over a gap.
Yeah, so you can see it starts to a little bit once it reaches a volume where it can really sense that there's gradient on the other side, but it really struggles compared to the smaller cells that can move through it. We also thought this was interesting because we didn't find a way within the restraints that we had to put this into the simulation. Also, once it reaches 300, that'll be the last value it records, and then it will shrink back down to the original size. Because since there is noise in terms of how it's distributed, on the off chance we see something like the first run that I did where it gets stuck, it acknowledges that there's possible variation between these values, depending on whether an unlikely situation occurred. So you can keep plotting it for the duration of the simulation over and over again and see the distribution of each of those speeds.
That's really nice, because the way I always did it was I had it do 10 rounds of the same value and then go to the next value. But it actually makes a lot more sense to do one round of each value and then repeat it so that you see everything first and then you get more resolution afterwards. That's a better way to do it.
Yeah, so it's been, I think, Andrew, you can speak to whether you were surprised, but I was personally surprised when we got this all up and working. It makes sense in hindsight, but this wasn't at least what I expected to happen.
Yeah, just the dips really affect the movement, and yeah, that really surprised me.
You know,
So
it kind of
sorry, go ahead.
it kind of makes me wonder, because the whole idea of the paper is that multiple cells would reach gradient faster and better, right? In the sense of so they have to develop some kind of communication between them to tell there is a gradient over here, and right, because a single cell here, it's having trouble to find that.
It's not, I think instead of the communication between cells, it's more about if a group of cells have found a way around the patches, the adhesion will pull the other cells that are not actually moving in.
Well, so that's an interesting question. I assumed that a bigger cell would do better. But if you had the same area, but it was subdivided into a lot of little cells, you're telling me it might do better than this. So it's not so you do want to explore a big area, but you don't want to be a single cell doing it.
So we don't have that here, but I will say I set up a very rough version of this simulation where I did blob initializer.
Oh.
I ended up and that code is what ended up being broken for a different reason, but tentatively it did look like a blob of cells adhering together with similar volume to the one big cell, ended up having a slightly easier time. There's one thing about the simulation, like I said, it's pretty freshly finished, that needs to be changed, which is that the region to the focal point that counts as you finding it is decided by where the cell's center of mass is. And for the really big cells, when it does get there, it actually ends up being, it's too much. So that needs to be varied before we put it up on NanoHub. But right now what you're seeing is that the same system, but I'm holding the volume at 25 and it's scanning through the noise. And it's stuck, so it's not really doing anything different. It ends up that any amount of noise in the environment, besides a very slight amount, is pretty brutal as far as chemotaxis goes.
I guess you could reduce the amplitude of the noise, right?
That's true. It's
set
actually
the parameter of the noise amplitude, the strength of the noise, as opposed to the amount.
Yeah, that would be a good addition. I'm actually a little right now the way that the noise is calculated is that you take what the chemical would be at that spot and you divide by 1.2.
But it's 20% noise.
Yeah. So which is significantly less than what they used in the paper, right? Because it was holes punched in the gradient.
Oh, well, if you're looking at the cell trajectory now, this is classical subdiffusion,
Mhm.
right? It sticks in one place, then it jumps to another well, and it sticks in one place, it jumps to another well. That's a very classic pattern of movement.
Oh,
It's called subdiffusion. It happens a lot. It's not one of the classic physics type diffusion laws, but it's if for example, suppose you had an egg carton and you had a bead or something in the egg carton and you shake it. Most of the time the bead stays in the same well of the egg carton, but occasionally it jumps into the next one, then it gets stuck there for a while, then it jumps to the next one. So you have two time scales, one of which it's stuck in one of them, and then what's the time it takes to jump out of a well. And so that's exactly what you're seeing here, which is actually a really statistically rather hard to characterize system, and one that you see quite a bit in reality. That's very nice. But I think yeah, maybe when you're doing the volume scan, instead of changing the target volume, you add another cell. You have all your cells be, say, 5 or 10 voxels, but you add one cell, you have one of them, two of them, three of them, four of them, five of them, and you see how they behave. My guess is if they stick to each other too much, it'll behave like one big cell.
yeah.
If they stick not at all, then they're all independent, doesn't matter. If they stick just enough, they may be able to find their way through, like Josh's slime mold, they may be able to find their way through the pattern by making filaments and then pulling each other back.
That's roughly the hypothesis that Andrew and I were working with. The idea that maybe the multicellularity side of things gives you the flexibility to still fit through the gaps in the gradient,
No.
but also the surface area to sense more of what's happening in your environment.
I have two questions actually. The
Mhm.
first one is, did you implement any kind of surface plugin on
Yes.
this?
Yeah. So
What happens when you put the surface higher? Does this, because right now it seems that it's very motile in the sense of,
right, the way that can people see my okay. So yeah, so we just did classic scaling of the target surface relative to the volume of the cell. And we tried to hold that ratio as we increase volume. So we did go ahead and do that just because otherwise the chemical field would be pretty aggressive in terms of tearing the cell apart.
and the lambda for that is also high to keep
It's
that
not
in
that
place?
high. That was kind of a trial by error situation where Andrew and I looked at it and we were like, what seems to be a value that results in appropriate behavior? I think it's actually somewhere between 0 and 1. Yeah, it's 0.5. But it does make a difference in terms of that or not having it there at all.
Well, the second question is, there is no way for the cell to ignore a patch, then there's no way for the cell to, well, I guess it depends on the size of the patch, right? But we were seeing that even like smaller patch, the cell didn't want to go there, even the shape was changing.
Yeah, I mean, that makes sense, right? Because so basically, if you end up with a wall of patches in front of you, then you're saying, okay, the energy cost for CompuCell of moving here is roughly the same energy as moving back. So you're just going to move stochastically. And then there's a chance that you'll move past it, but it's pretty low. So I think that given the way chemotaxis works, that behavior makes sense. I will say one thing that we removed because we wanted to see how this behavior played out normally is that we took out persistent migration, which was in the normal paper. And that's something that would get you over a small hump, right? If you're moving forward and then that's something that we could put back in, but for the sake of getting a whole new simulation working that wasn't a replication, we wanted to start with the core aspects.
Yeah, it looks very nice.
I think that's also interesting. I mean, here, right, you change the amplitude of the holes, and you should see that for small enough amplitude, the thing doesn't pin. For bigger amplitude, it pins more and more, which is what you're seeing here. So the time you spend jiggling around and the interval between jumps, between pinning should be longer as you make the pinning stronger. And then if you, as you say, if you introduce persistent migration, you'd expect that there's going to be some, as you increase this persistence, you overcome that pinning. And so again, you should see it's a pretty rich, if you're thinking of it as a physicist, there's a pretty rich phase space of this. There are a lot of different behaviors with transitions between them as a function of these parameters.
So this pinning, it's what I think is going on is that every 2,000 steps right now, what we're actually scanning through the noise. So then by at the the next scan, the noise increases by 1%. But what happens is that it goes into a function called initialize seasons, where it basically takes it reinitializes all the patches as opposed to taking the existing patches and adding more to them. So I think what's going on with this kind of binning is that by changing the distribution or changing the spatial distribution of the patches where the cell originally is, those patches are changed and moved differently, so it gets freed in that moment and then finds another place to be stuck in.
That makes a lot of sense. I mean, the fact that in a sense, it's more interesting because if it just got stuck and stayed stuck forever, you wouldn't see much. So the way you do it, it's nice. I'm surprised that the pinning is so strong.
Yeah.
And so it'd be interesting if you reduce the depth of your holes, make it 5% or 2% or 1%, say 20%. What level you unpin at?
I mean, I don't think that would be too hard to change. Where's our that was in the initialize seasons function.
Yeah.
Right, I divided by self.noise, so. Yeah, so does anyone want to pick a value?
1.05.
Okay. And what do you want to scan?
Well, whichever with the noise, I guess, if that's what you. Okay, so it's not pinning at all here. I hung up once. Now it's got hung up there. Now it's stuck. That's a really pretty demo. All three of the talks are really pretty demos. There's so much fundamental. I mean, it's also interesting. This is also interesting from the point of view of a sort of an optimization and targeting problem. How do you find a target in a noisy environment? It's really stuck.
Yeah, the experience with the cell was roughly our experience debugging it initially, but thankfully we managed to make it through. But yeah, so that's
saltatory program.
Yeah. So I think maybe I mean, it might delay our uploading to NanoHub, but it might be worth adding in a similar selection panel for the depth of the noise.
And the strength of chemotaxis.
I actually think that the strength of chemotaxis is pretty sufficient. Adding in persistent migration, if I had to pick between seeing lambda chemo and persistent migration, I would actually, but we could do both. But I think that those would have to be things that maybe get set as initial conditions. I think as far as parameter scans go, this works out pretty nicely. No. Anyway, so that's what we had to show.
That's great. I'm just empathizing with the stuck cell. It's amazing how you anthropomorphize these things.
Yeah, I felt like I was looking into a mirror for a little bit there.
I want to get forward, but I'm stuck. Yeah, that kind of pinning, actually, we published many years ago, really a long time ago, 20 years ago. We published a paper doing image analysis that way, where you had a pattern that you wanted to match that was noisy. And so we used the pattern as basically a chemoattractant and then put cells that would fill out. So it's a little bit, Josh's talk, right, is about optimization in the sense with noise. But yours is also about optimization of noise. So here you say, okay, I'm going to grow a cell out. It'll pin on the boundaries. It'll come up with some approximate boundary. And it winds up that you could do some image segmentation. These days, it's not the way people mostly do image segmentation, but we actually did image segmentation using compute. So we were analyzing X-ray tomography images, which are noisy. And the question was, how do you reconstruct the boundaries of domains when the image has a poor signal to noise ratio? And so we're actually combining the two approaches that you presented and Josh presented a few minutes ago. It worked pretty well. We didn't push it. The student moved on, and so I think it's unfortunate because I think the methodology. Veronica Gruneson, biophysicist who was originally from Brazil, and then studied. She was briefly here, then moved, finished her PhD in Utrecht in the Netherlands, and is now a professor in England. She independently developed a methodology like that she pushed pretty far for doing image analysis, segmentation problems. So there's a lot one can explore. That's great. Well, Alex, I don't know whether you have anything you want to add, but you're the last, so you get to close us out.
I didn't really make any like substantive changes to my presentation that I gave last week. A lot of the comments, had I like acted on them, would have turned it into like a C++ parallel programming lesson. I guess I could kind of go into that for like a few minutes.
Sure, that would be good. Clearly, your talk last week intrigued people. There were a lot of questions and comments. So if you want to show us that, maybe just answer some questions if people have them.
Yeah, I can talk on it in a few minutes and just answer questions.
Great.
So just to remind people, we have this field extraction in CompuCell that we have to pull out the data, all those data structures and like where cells are and all these chemical fields and whatnot, to visualize it, because we can't read data because we're people. Readable data formats are always a contentious debate. So this is important, but it's really slow right now. And it's slow because it's all these simulations can be very large. We have various data formats, various things we have to look at, cells, their borders, chemical fields, and we're doing this all serially. So to look at some of the problems that actually come up in the code. So if this was just taking each, so this code here is from CompuCell, just rendering each voxel what cell type it was. Because it's my understanding there's no way, like a cell doesn't know where it is. We just store the giant 3D field that maintains all the data. That's a different topic. But so when you look at this, we're just iterating over the first dimension and the 0 dimension, or X and Y. This is for a 2D plane. If it's not, if there's no cell there, if it's medium, then we just skip it because the underlying library called VTK makes it black for us. And then if it is a point, if it is an actual cell, we have to find all we have 4 corners on our square, right? So we have to add all of those to our array for the boundary points for each voxel. Insert that into our array, so we have a lot of VTK data structures. This one points stores the Cartesian coordinates. Then this cell array actually is this kind of like metadata structure where it says each point in this points array is 4 individual data bits, and then this is how they're related to each other. So it says these 4 in order are like one larger thing that we need to know about. And then the cell type array is where we actually finally store the information we care about, which is like the data typing. So it's great, it works, but there's issues with this. Like if we want to run this in parallel, so we have 2 like threads performing execution, like on, say one's doing the top half of this field and the other one's doing the bottom half. When you're doing, so think of like even an array in Python, when you're doing an insertion, the framework, the library underneath assumes that no one else is trying to do that at the same time. Because maybe you have to create, you're adding new things, you're changing the data structure, maybe it has to do some memory allocation. That is not thread safe. It's called a race condition. Because you have, 2 things trying to access the same item at once. Like what if you're trying to you can't share a pair of scissors. That doesn't end well. And then a really subtle thing is that this code here implies ordering. So that so you see here, so that we always guarantee from this code that we will update the points array and the cells array and all of our data structures we updated together at once, in that same order every time, which actually matters when we try and have multiple things executing at once. So we have to think about these things. And then, so I explained OpenMP, all this. So then the way, so to a quick example, so threads work in that they, so you have a process, which is, so it's like a higher level thing than a thread. So process has an execution stack, memory, and state. And then a thread shares that memory between other threads, but then it has a unique execution state. That's a very operating systems topic. So then when we have to deal with this kind of like inherent ordering, and this like guaranteed interaction with our data structures, we need to either ensure that we're not doing this kind of like concurrent memory allocation and changing of data structures, or really we have to prevent that, but there's 2 ways to do that. So the first of which is with various contention. It's called contention, or no, it's the issue that comes up. Well, OpenMP has critical sections. You provide exclusion. we rely on the library or OpenMP we just tell it this area has to happen only one time. Only one thing can do this at a time. Here we're inserting our data points into a larger data structure. And this is doable without OpenMP. There's libraries for this, and you could do that, but that's just more effort on your part. Developers are just lazy, which is great. And then I don't have the code. No, so I'll change what I'm sharing. So the other alternative to this kind of race condition of allocating memory or whatnot, affecting these data structures in mutable ways, is just prearranging things. So if we have this lovely pointer, we know how much data we're storing into that structure. So we can pre-allocate all of that memory because it's shared. And then each of our workers has an assigned chunk that it can then write to. So we don't have to worry about collisions or pointer error fun times. So that's, I think that's a lot of the basics. Did people have like questions on whatever?
It's okay. I know there are people, this issue of the difference between threads and processes was something that people were interested in. Issue of collisions. I mean, I know you talked about the sort of the time value of these things. That was interesting. That you made a comment which I thought was quite interesting, was that because you don't know what the configuration is, it's hard to optimize.
Yeah. So there's all of our simulation fields are just squares, rectangles, either one. So we could render the whole field. And then that makes it easy programmatically because we know how big it is. We know we could just assign each worker a chunk and it could do that bit. And that makes sense. Here we go. But it becomes an issue. So if normally we're only the serial version is only having to render this 20% of the area, and then we're suddenly rendering all of the area, even though we're doing it with a lot more workers, it's not faster because each of them is still doing the same amount of work as the original. And this is, so it's like a trade-off. Do we which there's, I mean, and most simulations presumably look like this lowercase, right, or bottom case rather than the whole area being full of cells. So this is the case we have to optimize, but then that means we don't know ahead of time how much area we're looking at to render. I mean, especially when you start. So some workers may see, they still get defined chunks, but some workers will see no cells to render, and some will see a lot. So we have to do that work, collect all of the data, decide, see how much we actually have to do, and then we can finally put it in the VTK data structures. And then that problem always existed for things like cell borders, because we literally have no idea what those exist like.
Well, I was just looking at the picture you got on the top. Cell borders are problematic because you have a higher density of borders. Even though you have cells everywhere, there are more cells in the middle. So the density of borders is higher in the center than it is at the edge. I mean, this is the kind of, in a sense, that this is the kind of thing that we face a little bit when we do things like diffusion. Our diffusion is done on a square lattice with a fixed discretization. If we used an adaptive mesh, you could adjust the mesh density to suit the gradient of the chemical. You put a high mesh density where there's rapid changes, and the low mesh density where you have slow changes. As I think you pointed out, somebody pointed out in class that if you have a fixed, no, somebody, it was a seminar. If you have a fixed mesh, whether it's in time or space, you have to adjust that mesh to be small enough that it suits the most difficult thing you're trying to solve, whether it's temporal or spatial stepping. And so then you do a lot of extra calculations in the regions that are easy. And so that's a little bit what you're running into here. On the other hand, doing adaptive meshing or adaptive thread allocation here is complicated, as you point out.
Yeah, it's doable, but you have to then, you're supporting two different things.
I suppose the trade-offs are depend on the particular problem, whether it's worth doing the extra work or whether the things you're wasting are essentially not that important. And then the code complexity, because using a full fancy adaptive mesher is hundreds of thousands of lines of code. Of course, if you could borrow one from somebody else, it's okay, but you're still sitting on hundreds of thousands of lines of code to do mesh. The using a fixed square lattice, it takes 5 lines of code to do the whole thing. So it's interesting to see that. Were there any other questions? I know there were other people who had some technical questions. I think everybody was intrigued. I haven't ever done OpenMP. I recognize it, but I've never done any OpenMP coding myself. Done some MPI coding the old days, but not OpenMP.
Yeah,
How many people? Having to do explicit shared memory makes life a lot easier than having to do explicit message passes. When you do this kind of thing with OpenMP, with MPI, you have to do these buffers. Every time you write to a buffer, you have to stack it, then you accumulate a stack, and then you send the messages across. It's a bear. Here you still have to do synchronization and locking, but it's not. But at least you don't have to explicitly stack the messages you're going to pass. I don't know how many people have done parallel coding as part of their classwork.
Yeah, I had, because it's part of the curriculum, but not so much in deep. It was just like an exercise about starvation, and you've been there, Alex. You know exactly what I'm
the basic
talking
stuff
about.
is like manually create like 2 threads that work together.
Yeah. Do you have any hypotheses about why the 2D is so worse compared to the 3D? I know that the amount of data is probably one of them. In 3D, you have way more data, but do you think the VTK deals with the 3D more naturally than it does with the 2D?
No, VTK doesn't really care about that.
Okay.
Because it seems pretty. So all my measurements were just the CompuCell code. So I didn't look at if VTK is better or worse. In either case, with the 3D ones being a lot better. It's usually because we were already having to render so much more.
Yep.
It was a lot easier just to. I mean, the work per thread was already was a lot higher, so you get more benefit out of that parallelization. Because with the 3D field, you don't even know. You can kind of like optimize it even where like, what if a cell is only inside, like it's never touching medium, which means that we would never really see it. So you get more benefit from the parallelization.
Do you see any benefit? Oh, go ahead.
No, I was just going to say one of the things at the beginning. I think at the beginning of class we loaded we just ran without looking at the details that Furkan's old colonic crypt simulation. Did we do that here? I don't remember. It's embarrassing. I don't remember. I used it in the Computational summer course, and and we ran it, and it was a mess. The visualization was a total mess, and we realized that some of the shortcuts that player uses in 3D rendering are not right. And so if you turn on the surface rendering, show the cell boundaries, it does it correctly. But in that particular simulation, some of the assumptions about the geometry, about what needs to be rendered and what didn't, were wrong for that geometry. And so you'd run the simulation, think cells would just disappear. Visible cells, I mean, clearly on the surface, would disappear because the algorithm it used to decide what was going to be rendered, what wasn't, hadn't thought about the U-shaped geometry that it was addressing. It had assumed things would be spherical blobs. And so boy, was that embarrassing as a demo, I'll tell you. 85 people, they say, run this demo. And they say, where did all the cells disappear to and why? And of course, on the fly, I didn't realize that if we turned on the surface rendering, it slowed things down a lot, but it actually stopped. It turned off the optimization. And so then you weren't the victim of the algorithm and the bug in it. But boy, that was, I don't think that's been fixed, by the way. I think Computational still what it tries to decide, says, okay, the cells are in the middle, so you don't have to show them. That algorithm seems to be, and I don't know if it's a VTK algorithm or if it's a Computational algorithm, but it's buggy. Because in principle, in 3D, in Computational 3D Player, you could say, make these cell types transparent so that you should see what's inside of it. It doesn't work. Again, if you render surfaces, you're all right, but then it's slow. But the optimizers come. And it's exactly what you say, that they thought about it for a specific situation, and then somebody used it differently. Sorry, Joel, you had a question. Go ahead.
No, I was just, what do you think, Alex? Do you think this new engines for video game could help in the sense of if we change from API and go towards that direction, or finding an API that can pre-process data and just use the engine to, produce the image?
Right. So there's two different things that are happening here. So we're giving data to VTK to render, and I don't know, maybe that could get faster. I don't know about that, but the issues here are that we have to marshal our data into a format that VTK understands, and that will essentially need to happen no matter what rendering backend we're using, unless we always keep our data in those formats, which is viable, but we have to rewrite all of Computational, essentially. So there's no way, changing rendering engines might help, but it wouldn't fix the problem here.
Yeah, I was just thinking that maybe there was something new in the new engines that could make stuff happen faster. But
I'm certainly not an expert on game engines and how they do rendering. There's a lot of them out there. And they change a lot. And they do really impressive things. But all those things, like those textures and lighting sources, are already in those formats that the game engine expects and is
it has to be
optimized to work on.
Other comments or questions? Well, thank you for going over the details there. That's exciting.
Yeah, no problem.
And I do appreciate it. So where did you learn parallel programming? Was there a class you recommend to everybody? Or did you do it on your own, or a class you would say avoid? So that it's not the way to learn it?
I'm the initial, how to just think about parallel programming. I mean, it was just undergrad operating systems cares about this, because just threading in general is an operating systems concept. And then when I did programming for a couple of years, working on web applications, you're at least back-end web applications, you're inherently threaded. So you have to care about these kind of things, especially like modern applications only exist as your C#, Java, Go, all exist as threaded programs from the out of the box. But like specifically for MPI and OpenMP, there's a CS and cross-listed engineering class by, I just took it last semester, that goes into this, like high performance computing, that's all it is, that goes into both of these topics. So just from the ground up, how to do OpenMP and MPI programs. And you get to have fun on the supercomputer.
I mean, we run CompuCell on the clusters here, but we don't really take advantage of the high performance computing side of it. We just use the fact that there are a lot of boxes, so we run a lot of replicas.
Yeah, you're not doing
everything is embarrassing parallel. So you run 1,000 jobs at once, but you don't try to couple them.
I mean, you need both, because like if you're doing, you're doing these kind of there's randomness, so you need to do multiple runs of the same simulation, and then maybe you want to do parameter sweeps. So you're running, you have to run lots of copies of a simulation, but then you can scale in multiple directions.
But sometimes you really want to run one big simulation. Then you have to. If it takes a month, you really would like to speed it up. The wall clock time per simulation may matter. Even if ultimately, if you're running 3,000 simulations, whether each simulation takes 1 minute, or whether you run them serially or in parallel, it takes the same amount of time. But I think it's interesting. So anyway, if you have a recommendation for people who want to learn this as a good class, that would be. I'm sure there are people in the room who would be interested in it.
Yeah, the HPC class was pretty good, although hopefully it'll be back to in-person lectures, because when I took it, it was pre-recorded videos, which isn't that great. But like the guy who teaches it, Sterling, is like the guy in the HPC field, like knew Cray and everything. Or there's always tutorials for everything out there on the internet.
But Thomas, so Thomas is the one who teaches it. That he should be.
Yeah.
I know him as a colleague, but he strikes me as somebody. He's extreme. He's been in the business of HPC since before there was HPC.
Oh yeah.
He's a real old timer on this.
He's got pictures to prove it too.
He's a legend.
Yeah, he's a great guy. I like him a lot.
Yeah, I'm interested in that course. That's something that, but just for the, having someone holding your hand when you're doing supercomputer stuff is great.
As long you got to know C++, that is a critical thing. But other than that, yeah.
I don't know if this is true, and maybe it's a little off topic for today, but one of the problems we faced early on, even with MPI, was that every cluster had a fundamentally different architecture. These days, is it Slurm or is it something else for job control? But the problem was that CompuCell was written to be something people could use locally. And every supercomputer had its own architecture. And so it was almost impossible to write a parallel version that would work on multiple machines. And I don't know if that's gotten, I know for very high performance computers, it's still very specialized. Just like like the problem with GPUs, right? If we write in CUDA, okay. But if it's some custom thing from Toshiba that uses their own special.
Yeah, it's my understanding that, so the point of MPI is that you have a library interface. So ideally, it should be like architecture agnostic. And from what I've seen, because in the HPC class, you were able to like run on a supercomputer, but then you could have like some kind of local MPI installation that would run the same code. And then like usually with the different supercomputers, you'll compile your code on that machine because they'll have some architecture specific library. Because I mean, like even not just like x86 versus x64 kind of architecture, but like networking setups, bandwidths, memory sizes, all these things there's magic optimizations somewhere in the NPI libraries. So each supercomputer usually has their own specific implementation that you compile against. But I've never heard of like it just breaking between, moving between.
I'm old. I mean, the old craze, when craze really were supercomputers as opposed to clusters, they had custom languages for writing parallel.
Mm-hmm.
And even between versions of the machine, they didn't support it. So it was a Cray X, and it was you wrote code for that. It was a Cray X +1, and you had to start over again because it was a new language. In those days, it was usually some extension of Fortran, but anyway, it's a long time.
People still write in Fortran. It's perfectly viable.
Well, I shouldn't say. It's dangerous to say it, but my old Fortran code, which is how what started out before CompuCell, ran awfully fast compared to where we are now. Given 25 years of improvement in computing performance, those old Sun Ultra Sparks were actually really fast machines for floating point operation. Of course, the difference was that code was extremely inflexible. It was all hard coded.
It was a trade-off between like abstraction and speed.
Yeah.
That always happens.
And so, I mean, but it did surprise me. It was a long time before CompuCell was as fast as my old code. I said, after 15 years of computer improvements, it really ought to run faster than
well, the core stopped getting faster years ago. Now it's all parallel. That's the only way to speed it up.
Okay. Well, thank you, everybody. I really appreciate you showing up during exam week. I appreciate you doing the additional presentations and talking. And I think everybody did a great job. I hope people will take the time to finish these things and get them uploaded if they can. Please send me, give me your write-ups and everything else. And if you want to finish, let me know if you're planning on finishing any homework assignments so that I'm prepared to make sure that I grade them appropriately. I don't want to, again, don't worry if you've missed one homework assignment. The world will not end for one homework assignment. And thank you. If anybody needs to talk to me about anything during the remainder of the week, I'm around. I'm not going anywhere. And again, I just want to thank everybody for your participation and engagement during the semester. I really appreciate it. So I guess that's the official end of the class. So everybody gets it's not even 8 o'clock. So it's half an hour early. Thank you, everyone, for being here, and look forward to seeing you either in person or virtually as things go forward.
Can I ask a question just before we get? It's
okay.
For the final should we send all the documents that we created for the presentation and the document for the thing and the link for the NanoHub application, right? Okay, that's all that I have to ask. Thank you.
Okay, well, good night, everybody. Thanks again.